When it comes to learning about the cell cycle, there is a lot of confusing vocabulary to navigate. Specifically in this video, I am going to talk about the differences between chromatin, chromosomes, and chromatids. Right now, I'm going to go ahead and start by describing chromatin. So, chromatin is the form that DNA is in 95% of the time. In relation to the cell cycle, this is all of interphase. When drawn out, chromatin can look like a snake or like a lax piece of string as shown here. Now onto chromosomes. Chromosomes are condensed pieces of chromatin, and this is important for mitosis. We'll get to mitosis later in the video. When drawing chromosomes, it is common to depict them as an X, as shown below. To compare chromatin and chromosomes in a different way, imagine a string of lights. How would you transport them from one location to another? If you leave them unwound, they would easily get tangled and are hard to move. This is like chromatin. If the lights are wound, they are easy to carry and transport. This can be compared to chromosomes. This is why DNA is in the form of a chromosome during mitosis. Lastly, chromatids. Chromatids are only recognized after DNA is replicated in synthesis, or S phase, and they are held together at the centromere. This is significant because when counting the number of chromosomes in a cell, the number of centromeres are what is actually counted. However, the chromosome has two halves, and each of the halves are called sister chromatids. Both of the halves are genetically identical. This is important because when a cell splits off in mitosis, you want to have two of the same type of cell. Now let's apply everything we've learned to the cell cycle. All of interphase is denoted in red. This takes up 95% of the cell cycle, and mitosis is the other 5%. A phrase that I use to help remember the form of DNA in interphase is chromatinterphase, because DNA is chromatin in interphase. The black arrow shows the direction of the cell cycle. Now let's talk about the last 5% of the cell cycle, mitosis, which is denoted in green. This is where we encounter chromosomes and chromatids. Now, let's fill in the phases of the cell cycle. First is G1, or gap 1, then synthesis, or S phase, G2, or gap 2, and finally M for mitosis. In G1, there is one strand of chromatin, shown by the blue. During G1, the cell goes through basic cell activities, such as growing, repairing its DNA, and other basic day-to-day functions. These can vary based on the cell type. In S phase, the chromatin replicates. I am denoting this by drawing two strands of different colors, green and blue. However, remember that both of the strands are genetically identical. These two strands are what will later make up the sister chromatids in mitosis. In G2, we still have the two identical strands of chromatin, yet the cell continues to grow and do basic functions. In this phase, the cell is prepping for mitosis, because mitosis takes a lot of energy. In mitosis, the cell goes through nuclear division through PMAT, also known as prophase, metaphase, anaphase, and telophase. Starting in prophase, the chromatin condenses to form chromosomes. Again, in my drawing, the two sister chromosomes are different colors, yet they are genetically identical. They are drawn this way for ease of seeing the products of mitosis. In the cell to the left, the two halves or sister chromatids make up one chromosome that is held together at the centromere. In a human cell, there would be 46 centromeres. Then, the chromosomes lined up in the middle of the cell in metaphase are pulled to different poles of the cell in anaphase and telophase, and finally split off to form two genetically identical daughter cells in cytokinesis. This is shown by the two cells on the right-hand side. This is the end of one cell cycle. Then the chromosomes will unwind, and they will be in the form of chromatin once more. Then the cycle continues. Now to recap, chromatin is present 95% of the time during all of interphase. Remember the term chromatinterphase. It is the form of long, stringy DNA. Earlier in the video, I described this as a snake or a string. A chromosome is only present during mitosis. It is the condensed version of chromatin. A chromatid is also only present in mitosis and is the name for each of the two identical halves of a chromosome. This is shown in the drawing to the right. Upon finishing this video, there are a few things that you should be able to do. You should be able to draw a picture of unreplicated chromatin, replicated chromatin, an unreplicated chromosome, and a replicated chromosome. Define the term chromatinterphase and explain its significance. Name the phases of interphase and describe what each phase does. Identify the form DNA is in during each step of the cell cycle. And lastly, differentiate between chromatin, chromosomes, and chromatids. If you find yourself not being able to complete any of these items, please rewind the video and review the materials. Thank you for watching this video, and I hope that you find it helpful.